And so you gave two separate talks at the conference. Can you give me just a brief overview? Because I know some of the questions that I have for you are maybe can overlap to both, but I just want to give the audience an understanding
Yeah,
of some of the main topics.
we have two very new trials which are in phase one studies, the ENLIVEN-01 and the TERN-701 drugs. 701 will be presented or was presented by Tim Hughes. I will focus on ENLIVEN-01. That's an ATP competing drug. That means in the traditional mode of action, but the pharmacodynamic and pharmacokinetic profile is much different from previous drugs. That means it's not cytochrome dependent. That means it has a different metabolism. Therefore, it's an ideal drug for combination, for instance. And also hepatotoxicity will be reduced with this drug. And it's not dependent on PGP, for instance. It's not dependent on the import-export enzymes in the cells. And it's a once daily drug which can be used after a meal. These are major improvements of the management of CML. But most importantly is the molecular profile of the drug. It does not inhibit many of the kinases which are inhibited by other multi-kinase inhibitors. For instance, KIT, SRC, PDGF receptors, FLT3 are not inhibited by ENLIVEN-01. Therefore, it's more specific against BCR-ABL. And second, it inhibits mutations very precisely, for instance, threonine 315 isoleucine, which are not inhibited by others. That means the combination or the alternative treatment with ENLIVEN-01 after or with the other drugs is feasible, and therefore the development of these new drugs is worth. And we support this very much in the phase one trial. Phase one had a typical design, 3+3, a step up design starting with 10 milligrams. Now we are at 120 milligrams. There is no DLT. That means drug is well tolerated. And the profile against heavily pretreated patients with efficacy is rather good. But of course, the main parameter in the phase one trial is tolerability, which is excellent with this drug. I like this drug. I treated personally 12 patients in this trial, and I have a very good feeling. But the data which we presented here at EHA are positive as well. We are now currently in the 1B phase. That means we support the previous findings. The 80 milligram cohort once daily is full, and we now randomize the patients without mutations between 60 milligrams and 120 milligrams. That's ongoing. And in parallel, we have a cohort for atypical BCR-ABL transcripts, which will be analyzed separately, and a cohort for the patients with the more problematic threonine 315 isoleucine mutation. Means we report here the efficacy of the step-up approach, the phase 1A cohort, and the tolerability at these doses. It's obvious that the duration of treatment is much longer than the duration of the previous treatment in these heavily pretreated patients. Therefore, I think the tolerability has been proven already, and we need, of course, go more forward in second line and into first line later to demonstrate the efficacy in the earlier lines.